There we go. Well, first I wanted to thank Adrian and Thomas for the invitation. I very much appreciate it. I can tell you at Loyal, we enjoy the Metabesity conference every year. So it's a really an honor to be here to tell you more about what we're doing at Loyal. Just as a way of introduction, we're approaching longevity drug development from, I think, a relatively unique perspective in that we're not primarily focusing today on humans. So you've seen this slide, and I know that a number of people have presented this as really the overarching narrative of why we're interested in tackling aging biology and developing therapeutics that specifically target the underlying biology of aging, with the hopes that we will be able to have impacts on multiple disease processes that occur with age rather than a single disease at a time. And certainly that's the underlying hypothesis that got me into this field and continues to make me excited about the opportunities we have to impact healthcare. But this is always shown, obviously, in the context of human health, where the incidence of major disease increases with chronological age. But this is also true of other species as well. And so I'm going to tell you a little bit about companion dogs. And so we see the same sort of trend, of course, occurring in companion dogs as well, in the cases where disease incidence and multimorbidity has been tracked in dogs through a number of great studies, predominantly by the Dog Aging Project, which I'll talk about a little bit later. And so the way we're approaching this at Loyal is that we all agree that the priority is to be developing therapeutics that target the underlying biology of aging rather than targeting specific diseases one at a time. But we think the strategy behind this is unique in terms of the way that we're approaching this problem. So, of course, thinking about this in the context of the development of human therapeutics, we know that human lifespans are relatively long, which, of course, by definition, necessitates relatively long clinical trials in order to capture enough data to prove efficacy, particularly in the context where you want to demonstrate a lifespan extension. Because of the amount of time that it takes, this, of course, is extremely capitally intensive to be able to do studies that are of high complexity and length. And of course, we've spent a number of days at this conference talking about the regulatory burdens and barriers to going after a true anti-aging claim. And we can talk maybe in the Q&A about some of the ways that we've been thinking about this from the animal health side as well. And so, of course, our solution is to do all of this work in companion animals. And we think that particularly the companion dog is the optimal species for doing this comparative aging biology work for a number of reasons. One, traditionally aging research is focused on laboratory animals, rightfully so, but living in very controlled and unnatural environments where they don't have the opportunity to experience sort of the varied environments, varied diets, and various experience stimuli that, of course, companion dogs living in our homes do experience. Dogs also have a very unique and diverse genetic structure and a wide range of phenotypic variations, which I think we can all appreciate when we think about different dog breeds, which allows for powerful comparisons within a species. Unlike other animals, dogs have access to a sophisticated healthcare system in which they are routinely monitored and treated for diseases in a system that's, only slightly behind humans in terms of complexity, in terms of the healthcare that's delivered. And ultimately, in terms of making a meaningful impact to people, understanding what we're doing in the longevity field, people have a very long-standing personal investment in the health and well-being of their dogs. So any sort of intervention that improves health span and lifespan in dogs will make a meaningful impact to the public at large. All right, so we know from our colleagues at the Dog Aging Project who have done a lot of the excellent pioneering work here in understanding what happens with dog aging, that the companion dog is truly an excellent reflection of human aging in terms of both of the mortality hazard curves in the way that dog populations survive out to well-defined ages. We know that they have common pathophysiological causes of death in many cases. The outstanding example of that not being the case is in atherosclerotic cardiovascular disease. But in general, dogs typically have the same diseases and die of the same sort of things that humans do, which is helpful in terms of unraveling the underlying aging pathobiology that's driving these diseases. Outside of also diseases that cause mortality, we know that dogs also have age-related changes in various functional domains that are similar to what's observed in humans. So in this case, we've asked pet parents to describe the changes they associate with their aging dog, and then we group those into various functional domains. And you can just see from these functional domains that a lot of these reflect the domains that we recognize also in human aging. And so it gives us a lot of power to understand the underlying changes in biology and their functional consequences and how that bridges across into other species. I guess most importantly, we know, at least for one aging intervention, that it has translated extraordinarily well between the rodent studies and the humans. So in this case, we know that caloric restriction studies in rodents generally are very powerful, extending lifespan. And given graded caloric restriction, you see graded increases in mouse lifespan. You know, a pioneering study that was done by Purina, examining a cohort of dogs from birth to death under caloric restriction or ad libitum feeding, demonstrated a remarkably similar percent increase in lifespan with 25% caloric restriction. So really demonstrating that these interventions also work well in dogs. So the way that we've come to this problem in this field is really the need that we feel there needs to be on more robust translational validation, right? So we have a number of studies that are ongoing that have given us a tremendous amount of information about the biology of aging. We know actually much more than most people think we know about the underlying process of aging biology. And that's, you know, manifested in the fact that, you know, by my count, and I'm sure there are more since I did this last count, we have 72 unique compounds that have been demonstrated in mice to extend lifespan. But what I think is very interesting is out of those 72, less than half of them have been replicated. And most of those have been only done at least twice. There's only 3 drugs that have been done more than 6 times in a mouse. And so there's an ongoing need for filling the translational gap in longevity science. And that is being able to provide robust and reproducible findings by both independent investigators, but also in a diverse set of organisms. And this is really where the genesis of Loyal is as a company built to start to fill in that translational gap and translate all these wonderful discoveries that have been made by the geroscience field into actual therapeutics. And so the core tenet that we work under is that we believe that companion animals are the fastest way to an FDA-approved longevity therapeutic. And that's really influenced by 4 main components. One, we can do our validation in dogs and ultimately commercialize in dogs. So we're actually not running the risk of having a species misalignment or a gap in terms of understanding. We're actually doing all of the work in dogs all the way through, which helps us to have great confidence that what we're doing in our early studies will ultimately translate. I think the most important part of this is that we're using lifespan ultimately as an endpoint. We're actually doing a lifespan study to demonstrate that these interventions increase the lifespan of a dog. And I think that's very important, powerful tool for using companion dogs to demonstrate these effects. And this is only, of course, made manageable by the fact that dogs have a much shorter lifespan than humans. So on a relevant time scale, we can actually perform a lifespan extension study in dogs. And finally, I think the accelerated regulatory timelines have allowed us to really move forward product candidates in a way that would not be possible, certainly in human medicine. This is highlighted by the fact that we have now advanced 2 candidates into clinical development in less than a year from initial validation in dogs to ultimately moving to clinic. Okay, so as that is the basis, I just want to talk about a couple of different concepts to explain a bit about what we're doing at Loyal. The first is this idea of leveraging the great phenotypic diversity that's seen in dogs. So one of the most commonly known phenomena in dogs is that they have a wide range of lifespans by breed, and this is very highly correlated with their adult body mass. So large breed dogs tend to die significantly faster than small breed dogs. And this is actually a very powerful tool for us to begin to look at the association of aging phenotypes by genetic associations. By understanding the genetics that drive the phenotypic differences in dogs, of which there's great pioneering work being done by Elaine Ostrander, we can begin to associate these aging phenotypes now with these associations for a number of different purposes. One, to understand the mechanisms that drive lifespan diversity in these dogs, to be able to understand the mechanisms of age-related diseases in these dogs. So certainly, as you would anticipate, the incidence and appearance of age-related disease occurs in slightly different fashions in dogs of different lifespans. To be able to also understand outlier dog breeds. So there's certainly dog breeds that don't fit the general pattern of falling along the body mass by lifespan association, which I think is really interesting to understand why some dogs are protected from that particular association. And then, of course, importantly, it allows us to have very precise selection of clinical trial populations. We can know from some of the great epidemiology work that's been done by the Dog Aging Project to understand what we'd expect our target population to look like when we start interventions at various ages. So as an example of this, we just completed something we call the Loyal Healthspan study. This was a cross-sectional proof of concept study in dogs of different ages and sizes. You can see essentially it was a comparison of young small dogs and young large dogs versus old small dogs and old large dogs, to get at the differences not only of the shortened lifespan in large dogs, but also to look at the age-related effects on various clinical markers like health-related quality of life, frailty, and I think importantly for us, as we're continuing our translational work, is to look at the predictive value of various proteomic biomarkers and epigenetic markers as predictors for health span as measured by quality of life and frailty. So we're very proud that the Loyal team was able to bring through 586 dogs over an 8-month period. And it really demonstrates, and these are companion dogs, so these are people's pet dogs. So it really demonstrates the speed at which you can enroll these target populations when you're working in the companion dog space. And we've begun to look at the data that was acquired from this study now that it's closed out. Very exciting. And over the next year or so, we'll be rolling that out for everyone. So this gets to one of the core focuses of Loyal, which is building the translational aging assessment tools. One of the things that we spend a lot of our time doing is both on the preclinical side and on the translational and clinical development side is building the tools that allow us to accurately assess the trajectory of aging in these companion dogs. And this includes things like functional models in our preclinical space to be able to provide accurate predictions. It also includes protein and nucleic acid biomarkers, allows us to do early target validation. And then on the clinical and translational development side, building the frailty and multimorbidity assessment tools, activity, sleep quality, quality of life assessments, and of course, quantitative biomarkers of therapeutic efficacy, which will be very important for us as we're doing these multi-year lifespan studies. So of course, we've heard a lot about epigenetic clocks and the changes in epigenetic the epigenome with age in the context of humans over the last several days. And a really pioneering paper by Trey Eideker's lab at UCSD demonstrated that you could also develop an epigenetic clock for dogs, and in this case, use it in a way that could synthetically map to the human epigenome. So you could actually build an accurate correlation of human and dog aging across the developmental landmarks of each species, which has been incredibly powerful. And so we've actually exclusively licensed this technology for use in dogs, and our computational biology team is working really hard at increasing our knowledge of and use of these epigenetic clocks by generating next generation epigenetic clock algorithms to be able to predict biological age in these dogs. And we've announced that we've partnered with the Morris Animal Foundation to explore detecting cancer in their golden retriever cohort using these methylation assays. Ultimately, we want to apply these methylation assays to be able to detect biological age. And so over the next several months, we'll be releasing into the public free canine test kits that will be used as part of a citizen science experiment, what we're calling the Loyal Pack, that will start to collect a large amount of epigenome data from companion dogs in the field. Okay, so the next part in terms of our key focus is the concept of the opposite of caloric restriction, which is, of course, caloric excess. Yesterday, many of you may have not known. In fact, I didn't know that it was Pet Obesity Day. And the sad fact is that the majority of pet dogs and cats in the United States are overweight or obese. And that has functional consequences on lifespan. As you may expect, this translates very well to our understanding from human biology as well. A very nice study by Salt et al. Demonstrated that overweight dogs, either male or female, died at significantly younger ages compared to normal weight dogs. So this is also demonstrating that excessive nutrition drives early mortality in dogs as well. And we are really interested in the association, particularly of insulin sensitivity and its decline. It's known that insulin sensitivity declines in humans, and it's been well described as being both an obesity and an age-related disease phenotype. But it's also been shown in dogs, particularly again in that Purina study, that caloric restriction in dogs preserves insulin sensitivity late in life. So we're actually very interested in understanding the consequences of insulin sensitivity with age in dogs. And this gets us to really sort of the core proximal cause of insulin resistance around the dysregulation of adipose tissue. So this is, of course, most clearly described in the context of obesity, where you get adipose tissue dysfunction, which leads into ectopic spillover of lipids into skeletal muscle and liver as a key component of insulin resistance. But it isn't just about obesity. We think this is actually a key player in age-related metabolic decline as well. And I just highlight one study here that did removal of visceral fat from non-obese dogs. A very small amount of visceral fat removal resulted in a profound increase in insulin sensitivity in these aged non-obese dogs. So finally, this gets us to the second core focus of Loyal, which I think fits very nicely with the core focus of Metabesity, in that we are profoundly interested in looking into metabolic dysfunction and companion dog aging, and really exploring the link between obesity-induced metabolic dysfunction and how that relates to the mechanisms of aging-induced metabolic dysfunction. And so just highlighting two aspects that we know are conserved, again, between dogs and humans. Dogs have a significant redistribution of adipose from subcutaneous to visceral fat, the same as humans do with age. That was described earlier last year. And dogs also have an age-related increase in lipid dysregulation as measured by triglyceride levels. So all of these aspects are the core focus of the company in terms of the therapeutic strategies that we're developing, and demonstrating the importance of maintaining metabolic health with age. Okay, so with that, just a brief introduction to the Loyal team. We have a really passionate and experienced team working on all of these problems from both the biology and the computational biology end, as well as from the clinical and operations side as well. So happy to discuss more about that. And just as a final slide, you'll note that we didn't talk anything about drug discovery, because what we do is translational discovery. And so where our drugs come from a number of different sources and partners. So if you have an idea that you think will be meaningfully impact dog health, we're always happy to talk, and our contact info is here. And with that, I will take questions.
Terrific. Thank you, Michael. I'm not sure. Dennis, did you just get punted by the line? Whenever you join us, that'll be great. Adrian, I don't know if you have any questions for Michael to keep the chat going for the moment.
Well, actually, a couple,
Oh,
and then one
here
statement,
here I
because
am.
Dennis is
Right.
here. So I just want to say, though, I'll take this second. I'm getting messages from a variety of different places. People do want to understand this game. All you do is go on Cadence, log in if you're not logged in. If you don't know how to log in, look at your last email from Cadence. All the way to the left, right underneath the little icon for a house, you'll eventually see a little beaker icon. Click on that. It'll say ECS, that's our Emerging Company Showcase. Click on that and you will be in the game. That's all. Dennis, thank you. Go ahead. I'm sure you have many questions.
No. Thanks, Michael. Thanks for that presentation. Just a couple of things that you would agree, or would everybody agree that kind of dogs are a pretty good surrogate for humans in terms of it? And so that's accepted, I would say. Mm-hmm. What I'm trying to figure out is if I had a compound that dealt with kind of metabolic aging biology or something, why do I need you? Why wouldn't I just do that myself? Why wouldn't I do that myself? And so I'm struggling with what I'm struggling with is, how are you fitting into the whole plan here? And where's it going? And maybe, but so anyway, why would I need you?
So I think the biggest aspect that we bring is the speed to be able to actually get to demonstration of a lifespan extension, right? So if you had a strategy of which you wanted to do that, you can't realistically do that in humans. You could demonstrate a Healthspan benefit.
No, but why wouldn't I be running these? Why wouldn't I be running the same trials you are in dogs?
It's all about bringing it to actually commercialize as well, right? So you can run the studies, right? We're not a CRO, but what we're ultimately doing is getting FDA approval for these therapies, right? So that's the biggest difference.
But right. So could you walk me through that? If I had a drug, right, that has some impact on metabolic disease related to aging, could you just walk through how's it work through approval?
So you would have to go to the FDA and open up an INAD, so it's the animal version of IND, to allow you to do development work. You'd have to demonstrate through target animal safety and pharmacokinetics that you have developed a drug, not in the laboratory setting, right? In terms of actually demonstrating the drug is safe and effective. And then ultimately you'd have to run a pivotal study in companion dogs in the field. So a laboratory study is not sufficient.
Okay. And so instead of doing that, I contract with you to do it for me?
We're not a CRO, so we would partner on bringing forward an animal indication for your drug. Okay,
so now, and that all works. Who owns it at the end? Who owns the intellectual property?
Yeah, that's part of the discussion that we go into it as partners. Yeah.
Okay, so you try to do a partnership with that person, and then you have a negotiation about who owns what.
Correct.
And what about the? Is there any evidence yet that the FDA is going to be kind toward your approach?
I can share that we've had a number of conversations with FDA. I'd be careful about saying, in terms of where we are on progress on that, but we've had these discussions with FDA on the Center for Veterinary Medicine side, not the CDER-CBER side.
And is the CBER-CDER side the one that's most important?
For us, it's going to be Center for Veterinary Medicine. They're the ones who regulate veterinary drug approvals.
Right, but and then how are we getting from veterinary drug approvals to human drug approvals?
They're completely separate processes. So if you wanted to bring the same molecule in you'd have to open an IND and run it on the human side of FDA.
Okay, all right. So ultimately, Michael, you're not going to touch humans.
Right now, our plan is to work specifically on veterinary medicine indications. Yes.
Yeah. And you don't have a sense yet of, or maybe it's too early to tell, kind of what indications you might think about.
Well, ultimately, we're trying to get a lifespan label claim, which I think would be a big win for the entire field.
Sure. And do you think that's too broad or not?
we'll find out. We're in the process of doing that right now.
Yep. And how have you been funded so far?
So we've been funded by venture capital. We just raised our Series A in the last several months, closed a $27 million Series A.
Good for you. Good for you. And how long will that last you?
You have to ask Celine that, honestly. That's out of my purview. I handle the science.
Got it. Huh. All right. Well, I think that, boy, it's a big idea. So congratulations on the progress, and wish you the best.
Thank you very much. Appreciate it.
Coming on. Okay, Michael. Good to meet you.
Thank you, Loyal.